Hello everyone, welcome to my channel. Today I will discuss glycolysis with you. What is glycolysis? It is an oxidative process that occurs in cytoplasm which converts glucose into two molecules of pyruvate and releasing an energy. This process is also known as M10-Meyerhof pathway. What are the types of glycolysis? There are two types, aerobic and anaerobic. When cells have mitochondria and oxygen, they will follow aerobic glycolysis. If either mitochondria or oxygen is lacking, will follow anaerobic glycolysis. Anaerobic glycolysis occurs in erythrocytes, skeletal muscles. Why glycolysis are very important? Because the first point is it is the initial pathway of oxidation for provision of energy. Second point, anaerobic glycolysis provides an energy to erythrocytes where RBC transfer oxygen throughout the body. They also lack mitochondria, so they cannot use oxygen to produce energy. Anaerobic glycolysis provides energy. It also provides ATP in skeletal muscles, even in absence of oxygen. What are the steps involved in glycolysis? There are 10 steps. First step is the phosphorylation. In this step, glucose, an important energy source that is needed by all cells and organs of our bodies, is phosphorylated by ATP to form glucose 6-phosphate by the help of the enzyme hexokinase. In this step, magnesium ions have essential role for the activity of the enzyme. Magnesium ions interact with the negatively charged phosphate group and promote a favorable conformation of ATP at the active site of the enzyme. The second step is the isomerization. It is the reversible steps where glucose 6-phosphate isomerizes to fructose 6-phosphate. Phosphoglucoisomerase enzyme helps this process. Third step is phosphorylation. Fructose 6-phosphate is phosphorylated by ATP to form fructose 1,6-bisphosphate by the enzyme phosphofructokinase. This step is the rate limiting step of glycolysis. The next step is cleavage. The 6th carbon sugar is cleaved to form two 3-carbon molecules, glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. This reaction is catalyzed by an aldolase enzyme. The next step is isomerization. Dihydroxyacetone phosphate is isomerizes to form glyceraldehyde 3-phosphate. This reaction is catalyzed by triose phosphate isomerase enzyme. This step is based on the acid-base catalysis reaction. What do you mean by acid-base catalysis? This is a chemical reaction that catalyzed by an acid or base. Acid acts as an proton donor and base is proton acceptor. The 6th step is two molecules of glyceraldehyde 3-phosphate are oxidized by the enzyme glyceraldehyde 3-phosphate dehydrogenase to form 1,3-bisphosphoglycerate. This is the only step in glycolysis where NAD+ is reduced to form NADH. The 7th step is the transfer of high energy phosphate group. That means 1,3-bisphosphoglycerate is converted to 3-phosphoglycerate and produce high energy phosphate molecules, that is ATP. Phosphoglycerate kinase transfer this phosphate group from 1,3-bisphosphoglycerate to 3-phosphoglycerate. The next step, phosphoglycerate mutase, catalyzes 3-phosphoglycerate to 2-phosphoglycerate. This process also involves 2,3-bisphosphate intermediate. In the next step, enolase enzyme catalyzes 2-phosphoglycerate to phosphoenol pyruvate and releases water molecules. So this is a dehydration reaction. This reaction is dependent on magnesium ions. In the last step of glycolysis, high energy phosphate group ATP was generated by pyruvate kinase enzyme to form pyruvate. This enzyme requires potassium ion or magnesium ions that bind to the active site of an enzyme pyruvate kinase. This is the reaction of glycolysis. In glycolysis, the energy yield from one molecule of glucose is 2 molecules of ATP and 2 molecules of NADH. So this is the overall process of glycolysis. We are coming up with the next part of glycolysis. There we will discuss about the inhibitors of glycolysis. If you like this video, don't forget to subscribe it. Thank you.